So cool. This is the first episode of Major Bag Alert where we look at finding the next potential multi-bagger in the biotech space or potentially a tech space. And today I've got a pretty cool episode. It's with Norman, who's a PhD. Um, or you're not a candidate. You've already got your PhD, right?
Yeah, I got my PhD about a
Uh,
year and a half ago.
Oh right, okay. And what was the field in? Like, and I've seen your published paper on ROR1, but just keen to like hear exactly what it
Cell
was in.
and molecular biology.
Nice,
Yeah.
big stuff there. So
It's a lot of words,
got
really.
got a guy who's arguably smarter than me in the cell and molecular biology space. And yeah, like look forward to
I
hearing more
wouldn't be
about
too
the.
sure about that.
You're too humble, mate. You're too humble. But yeah, like it'd be good to like delve into ONCT or as a ticker, but Octernal Therapeutics, and just sort of like hear your views in the company. But I've got a couple of things that we can sort of like rattle off from the beginning. So yeah, I guess just like we've covered like what you did your PhD in, but um, let's just like delve into your background a little bit and sort of the research that you've been running.
Yeah, definitely. Thanks for having me on. So I got my PhD about a year and a half ago
Yeah.
here in Philadelphia, just down the street from Penn.
All right. Yeah.
My research was focused on ROR1
Right.
or ROR1, whatever you want to call it. No one really quite agrees. Um, But yeah, so I was studying ROR1 within the context of chemo resistance,
Right.
because one of the really striking findings that we found early on was that in patients that either don't respond to chemotherapy or go through a few cycles of chemotherapy,
Yeah.
there tends to be an upregulation of ROR1 expression.
Right.
So that was really just a very striking phenotype, and the bulk of my thesis work was like teasing out why that happens. And apart from that, I worked on cancer immunology.
Cool.
So after I got my thesis, I transitioned into a postdoc where I studied some of the mechanisms that regulate the development of some of these pro-cancer immune phenotypes. It was really cool, but I've since made the transition into industry and joined a small biotech company
Oh,
where
nice.
we're still in stealth mode, so I can't really talk too much about what we're working on. But hopefully, it's going to be big, and it's cancer drug development. So yeah.
It's interesting stuff. So yeah, you mentioned like the ROR1 or like ROR1 pathway. Like I feel like we should, yeah, let's just firm up a way that we're gonna mention that. So we just go with ROR1. Is that cool?
Yeah,
All right.
let's go over all one.
Yeah.
Rolls up the tongue a lot easier.
Yeah. So okay, ROR1. So
Hello?
hey, yeah, it seems like Twitter space is a little bit pernickety. Cool. Like I've got you back now
Progress.
though. Yeah. So just before that, I was just sort of um, just wanting to like I guess delve into like the explain like I'm five kind of outcomes of your paper because as you sort of know, like there's a whole load of retail investors out there don't necessarily have like a um, can you hear me?
Yep. Can you
Yeah.
hear me?
Yeah, I can hear you. There's a lot of retail investor guys out there, like they say, like what is it, like 29% of total flows in the market right now are from retail. They don't really understand biotech because it can get like pretty technical. And I guess it'd be good to just get like your explain like I'm five, like what the outcome of your paper was and like how does that sort of feed into uh, nocturnal therapeutic sort of outlook for simtuzumab.
Yeah, for sure. I think it's very key to summarize findings in a very explain like in 5 way. Yeah, I think it's something that a lot of scientists don't really do a good job with, because I feel there's this disconnect between the data and what people actually believe in, and you can see that with like the coronavirus and the vaccines and all of that. Anyway, but so the general gist of my paper is, I'm sure a lot of people are familiar with how cancer therapy usually goes, especially for the harshest cancers, where like you go through chemotherapy and then initially you see a regression in the tumor and the patient's getting better, but over time eventually there's relapse and the patient doesn't really respond to chemotherapy anymore, and the response to that is usually giving them even more chemotherapy. So the general focus of my work was understanding why that resistance happens.
Right.
And if we can realize why that resistance happens, perhaps we can develop ways to, one, either prevent that resistance from happening or go after these resistant cancers in a way that is less toxic for patients.
Right.
So that's a general gist of my work. And so how it relates to ROR1 is, we found that patients that don't respond to chemotherapy or that stop responding to chemotherapy, they have high levels of ROR1. So that directly correlates with high expression of this receptor that normally isn't really present on just cells, normal adult cells. So now that we have that finding, the reason it's so relevant for nocturnal therapeutics is it opens up this giant pool of potential drugs that they could do combinations with.
Yeah.
Because if you have ROR1 as this main agent that's regulating resistance to chemotherapy, if you inhibit it, then you could be coming in with a lot lower doses of these very toxic chemotherapeutic agents and getting really, really good clinical results. So, yeah. I don't know if that was 5, that's probably like 12, but...
Yeah. It's all, you did a good job there. I think everyone else probably says the same thing. Yeah, so I think another like cool part you sort of mentioned, like going in with much lower doses, and sort of having a much better like adverse effect kind of or side effect profile is key, definitely. And I think like just taking a look at, even though like some of the data that we've got currently for nocturnal therapeutics, I had a look at the Velis Bio stuff too. Like you get the sort of general spiel that there's no major ADRs, and like without having a proper view of all of the different data points, it's kind of hard to like find that, I guess, that hidden side effect that the company doesn't really want you to know about, kind of thing. So I guess like based on the sort of research that you've done right now, and like I guess also makes sense that because you can come in at a lower dose, it's likely to be safer. Let's play devil's advocate and just think about the flip side, because like with chemotherapeutic agents, like you see like the memes where
Yeah.
cancer companies come out and then it's like, yeah, there's no major ADRs, but that means that we've got grade 4 neutropenia or something like that, right? So like what do you kind of see as like a potential off-site adverse event? I mean, that could happen. Like I know it's difficult to say when you're like looking at cell on a cell-based sort of level, and then when you go in vivo, like things go to shit, kind of thing. But yeah, it'd be good to just see what you kind of think.
I think one thing that we don't really talk about enough is RL1 is ROR1 it's a good target, right? Like it's highly expressed in cancerous tissue, very minimally expressed in most tissue, but the emphasis really has to be placed on most. There's still significant ROR1 expression in just a lot of different tissues in the body, like cerebellum has got lots of ROR1, different parathyroid glands have got lots of ROR1, pancreatic cells have got lots of ROR1. I actually have a small anecdote with this. So I recently applied to a job at J&J. I actually got the offer, but I didn't take it. But one of the things that came up during the interview
That's
was
just like a little humble, like you got to throw that bit of salt in there, humble brag.
I mean, I can't be having people think that like I didn't get the job, you know, like that's why I'm talking shit. You know? Like, nah, it's not that. But
Yeah,
so we talked a little bit about their ROR1 program. So they have this ROR1 T cell redirection candidate. It's an ROR1 CD3 antibody,
right.
and they discontinued that project. And I think if you actually go online, you can probably find the patent for it. But the reason for that was because they saw so much toxicity, and it was just not something that they could rescue. So you could get, the drug to go where you want. You could do whatever you want, but there's just so much toxicity associated with it. In my research, we had a potential ROR1 inhibitor as well.
Right.
And I mean, obviously in the paper, I'm not going to say this, but it really does have a lot of side effects. Yeah, so it's just one of those where like it works, but I think pending the results of some of these more inclusive trials, especially as the companies become more open with those ADRs, like you mentioned.
Yeah.
I think ROR1 is a very exciting candidate, but there is potential for significant side effects.
Damn. That's not what we like to hear.
But
Because
see, that's the thing, though. It's basically every candidate you go after right now, that's going to happen.
yeah,
I will say RO1 is a lot better than other things in this regard. Like you've got very successful therapies for things like EGFR, some of the BCLs and anti-apoptosis proteins and things like that. These aren't genes that are selectively expressed in cancer cells.
yeah,
Right, but just because they're highly expressed in these cancerous cells, you get that added selectivity. So I think RO1 is still a very promising candidate, but we just got to keep our eyes out for these potential side effects.
yeah. It's interesting you mentioned you said like the parathyroid gland, like it's highly expressed there as well. Um, so yeah, and then you mentioned like the pancreas too, and I guess yeah, it would probably be quite interesting to see if there are any sort of like off-site reactions there that could cause maybe some thyroid disorders potentially.
Yeah, for sure.
And then that
Um,
that that could be a big kind of yeah. You can see a lot of issues going wrong or things popping up over there. Like you got
yeah,
patients with like goiter and stuff like that just popping up. And for those who don't know what that is, that's like a massive swollen sort of neck with build-up of
yeah.
thyroid hormone. Yeah,
One of the recurrent side effects as well that I keep seeing in all the clinical trials, at least the ones that have published
yeah,
updates, thrombocytopenia,
yeah,
like a deficit in platelets.
yeah.
That's not great. I don't really know why that happens. And I guess I don't know enough about other clinical trials to know if that's like a common side effect of going after some of these targets, but I
Yeah,
think that's equally worrying.
yeah, yeah. I mean, with like the thrombocytopenia stuff, that's quite a common um, cancer drug side effect. So I think that that's sort of kind of expected. Like you have like the standard ones that you sort of look out for, and no matter how I guess selective the companies say they are, you kind of just end up with this sort of stuff.
Yeah,
Always.
I'm also very interested in what's happening in the brain. A lot of clinical trials in oncology don't always look at what's happening at the blood-brain barrier.
Right.
So if you have a good enough small molecule, for example, it's going to make its way over there, right?
Yeah.
And the question now becomes what's going to happen over there. Some of the work that we had going on in the lab that I was a collaborator on was looking at the role of ROR1 in the nervous system.
Right.
Because that's actually where ROR1 was first identified in mice back in the 90s, I think. It's like
Right.
a neuron factor.
Right.
What we found is that it regulates differentiation of neurons into like adult neurons.
Right. Okay.
And we think there's a potential link with Parkinson's disease as well, but the data for that isn't really published yet, so I can't really comment on that. And I left the lab, so I'm not really sure what's up with that data. But the one really cool thing, I guess not really cool within the context of like disease, but cool within the context of science, is within cells, one of the main proteins that interacts with ROR1 is the amyloid beta precursor protein. Amyloid beta, that's the main causative agent for Alzheimer's disease. I'm sure you've heard, and a lot.
Hello. Damn. Hey, dude. Sorry about that. I don't think there's a time limit. I thought that it was like a pretty much a because I've been in a couple right now where they kind of just run, but
They just keep going.
yeah, it is what it is. I think because it's still in its beta stage kind of part, it's still a bit, yeah, but
Yeah, man, that work in progress.
go on Twitter, man. What are you doing? We're trying to educate people here, and you're fucking with our shit.
But yeah, no, if you could tell me a bit more about like your background and how you got interested in ONCT and RL1 and
So
your expertise and stuff.
yeah, like well, for me, like I'm a pharmacist from a background like perspective. So I registered as one in like the UK. It's nowhere near as prestigious here as it is in America, but I think that's just down to the fact that you guys have to do like a 7 year degree. But I think that's all down to money and stuff like.
I think every medical profession is that way. My brothers are one of my brothers is a surgeon up in Scotland, and my other brother is a PhD postdoc in Birmingham, and yeah, it's not as prestigious in the US. I mean, on the flip side, you get free healthcare. So.
Yeah, yeah. Like that's a big plus because like I mean I've been watching a lot of Grey's Anatomy recently and like I keep seeing these guys just like in hospital and they're worrying about bills and I'm like shit man like I don't want to have that.
It's crazy now. It's crazy. I'm pretty sure I found a paper on this, but I need to go find it. But there's a correlation between like stress as a result of worrying about bills and finances and actually worse disease like clinical outcomes.
Yeah, like I'm not surprised. Like you can't get sick. So if you get sick then you go bankrupt. Like
I can't afford to die. Like, it's like $20,000 for just
Mate.
throw me away. Just put me in the bin.
Literally, it's probably why CrossFit's like blown up in the US because it's like, all right, okay, I really need to be on my fitness shit because otherwise I'm gonna get diabetes and then I'm stuck with like taking insulin, which is just hiked non-stop for no reason, even though it was made like back in the 1900s and it's been off patent for God knows how long. But yeah, that's another story. Yeah, with the yeah, with the nocturnal therapeutics part, like for me, um, it I run a Twitter scraper on like the guys that I follow, like the biotech guys. So I use that as like sort of my way to kind of pick up stocks, just because I don't have all the time to sit there trawling through like the XBI or IBB or anything like that, or look for all these like small cap or micro cap stocks. And then it popped up, and I was just like, this it seems pretty interesting. And I saw like the Velos Bio buyout, and that was what for like 2.75 by Merck. Then I was like, okay, this is pretty
What
small.
what do you think about that? Those read. I don't know if you read it about how Ontario was essentially doomed because their candidate is not as potent as Velos Bio, and the whole rationale, which I thought was a bit silly, is that there's a reason why Merck went after Velos Bio instead of ONCT, which doesn't really make sense. What do you think?
Yeah, I mean, see, I've taken a look at the data. I've actually got the papers up here, like on my Google Sheets right now. So yeah, let me bring them up so we can, instead of me just relying on my pea brain to remember all the small details, uh, I'll actually look and read out the sort of information that we've got. So, in terms of efficacy, like just comparing, and I mean, like I know it's not a fair comparison because you're sort of comparing apples to oranges, like different patients, yada yada yada, all that sort of crap. But and I think what was also interesting, like in terms of that comparison too, I think it's kind of difficult just because the patients were on different therapies, right? So like the Velos Bio guys, they like I'm just looking this all up. The prior systemic therapies they had were hematopoietic stem cell transplantation, CAR T, natural killer cells, and then you had a BTKI as well, and then you've got some Tizumab's trial, and over there you've got like let's take a look. Pre-treatments. They only had two prior treatments, so it's kind of vague. In terms of like efficacy, it seemed like to me, Oncternal was trumping it, and I was actually reading a Seeking Alpha article where they seem to produce like better results. Like when you compared, and I can't remember off the top of my head, was it in MCL or CLL that they had better results? Um,
So this Ontario.
yeah, if I remember right, let me just double check because I had this down at the time, and it's just like really should have had this just off the
Velos
top of my
Bio do
head.
MCL.
Da, da, da. Key takeaways: no efficacy in CLL. Yeah, that was it. So in Velos Bio's trial, they didn't have any efficacy in because they tried it in a bunch of different cancers, right? And they showed efficacy in MCL and diffuse B. I think it's B cell, diffuse BLCL. Can't remember the thing off the top of my head. What is it? Before I make a fool of myself. Diffuse large B cell lymphoma. Um, and Oncternal had efficacy in both CLL and MCL. That being said, like they didn't test out all of the drugs as well, and also at the same time, Oncternal. I think Velos Bio wasn't a joint therapy trial,
Yeah.
whereas Oncternal was used with ibrutinib. So
I
not
think I think I don't know what the. I'm assuming they've got a patent for that combination now as well, right? Don't they?
uh,
Because
Oncternal.
theoretically, Velos Bio could just go after the same combination. Because if you think about the reason why it works is you get compensatory feedback through ROR1 when you inhibit BTK. So
Yeah,
if you go after both ROR1 and BTK at once, then you get synergy. So
yeah.
yeah, very interesting stuff.
Yeah, I don't. Yeah, they haven't got a pattern. And I don't. I guess like I think in this case, I don't think it's a big problem, just because like you've seen in quite a few, um, sort of like big cancer drugs that have come out like recently. They don't really have like a patent for a combination as such. Um, and in a lot of cases as well, like they do use like even older drugs that are potentially not necessarily on patent or anymore or are coming to sort of like the end of life. They kind of just use them anyway. So I think the differentiation is enough like for the single sort of therapy and how it reacts with other drugs to be enough of a moat. as it were. Because I think it would probably be quite maybe like you're just sort of adding time to development if you're going to look for a way to patent, I guess, a combo therapy when you kind of don't. Like, what's the benefit in terms of return on investment? Like, are you going to get that from that?
I wonder what that looks like in terms of AbbVie and Johnson, the people that make ibrutinib.
Yeah,
Um,
yeah. Let's
but
see
yeah, I'm
actually.
just thinking in terms of, I'm seeing a lot of rumors about acquisition of internal.
Yeah, actually, like you bring a good point up there because I can't remember off the top of my head, but I know recently they were passing some legislation where it was sort of essentially trying to stop like the big dogs kind of merging together in terms of like big pharma. So you see that as sort of like a catalyst to push M&A further down the chain into like the small and mid cap
Mm-hmm.
type pharma companies. So, like, I mean, that's always been in the case, and I think like pharma probably got like quite a bad track record, right? Like buying up companies a bit too late when they could have jumped in a bit sooner and got a much better deal. But yeah, that could definitely like push this sort of stuff. Um,
Yeah.
there was like one question that I think Peter
Oh
like
yeah, I saw
brought
that,
up.
and I was going to reply to it, but when we got on this call, and yeah, it's a great question. Do you want to read it, and then
Yeah, let
I'll answer.
me bring it up. That was quite a good one. Oh, I think actually we've got Peter on the call, so we could probably just
Yeah,
get him to.
yeah. If you can hear me,
Yeah,
does this work?
Yeah.
Sure. Hi. So my question was, um, part of the thesis of the paper was that inhibiting ROR here is going to upregulate P53. That
Yeah.
these are not wild-type P53 lines, right? And if they mutated or they P53 null, and so I'm sort of puzzled by that. And
So
whether you looked at it in the context of
uh-huh.
lines that were P53 wild type versus mutated versus no.
Yeah, so we have data from other P53 wild type lines, and we don't really discuss this in the discussion section of the paper, but that same phenotype isn't really seen.
doesn't
Do you
go
hear me?
to it. Yeah, I can hear you.
Yeah, so these are P53 mutant, not P53 null lines. So they've got very, very significant reduction in P53 expression, but it's still low and it's still functional. So what we're seeing is, we're actually seeing two phenotypes. One when you knock out RR1, well, actually knock down because we didn't do a knockout, and one when you inhibit the protein. Right, so you can already imagine that if you knock down the protein, you're going to get significant or more feedback. So the phenotypes are going to be a little different as opposed to just direct inhibition with our drug. So anyway, what we found is we get increase in P53 activation. What that means is you get more P53 going into the nucleus and activating transcription. As an added bonus, what we found with the inhibition with strychnine is we see an increase in P53 expression. So that's the cool finding. It's not really, so I want to say RR1 inhibition always increases P53 expression, but it seems to increase P53 activity. So I think that's a little bit of that's where the discord was with that question. Oh, great question, Peter. Thank you.
Cool, man. So yeah, I think apart from that, like we've got, all right, we know we've got mantle cell, we've got chronic lymphocytic leukemia. Those are the sort of targets that right now simtuzumab is being aimed at, and then you've got another catalyst for HER2 negative breast cancer, which aptly works pretty well with your chemo resistance paper. So looking forward to that. Like we've seen a bit of a run up in the stock recently, and I think it's trading like up at nine right now, isn't it? If
Yeah,
I
yeah.
it's yeah, it seems to be getting a bit of a buzz around it. Be interested to see who's holding it institutionally. But right, I think like the main sort of thing here is like let's. I know you're a PhD in cell biology effectively, but let's go and put like our investors' hats on. If we were to put this out as a therapy and assume that what we're in phase two right now, get positive data, and then we move to phase three. We've probably got what maybe like another 2 to 3 years maybe max before we start hitting around like the FDA PDUFA type scenario at push. But what do you reckon like we could start seeing in terms of numbers? Like it'd be good to just get like a round robin of what everyone thinks we'll start seeing in terms of revenues.
Yeah, I just want to preface this with I'm a terrible biotech investor, right? Because I just get excited about like technologies, right? Like if I see a company is, I don't know, making some ointment that's going to cure cancer, and they've got a terrible paper that seems to work, I'm on board. It's just cool to me, you know. And it's not a very effective investment strategy.
Yeah.
So I got into ONCT very early on because it was my work essentially.
Yeah.
And I just really believed the potential for it.
Yeah.
I think you've got a spot on. I think the way the data is looking, I think at least with MCL and BL and a couple other cancers, I think within a couple of years we're going to see tremendous revenue. I'm also very intrigued by some of the other ROR1 companies. I think some of those have got more potential. Like I mentioned NBE before, but
Yeah.
I'm just very intrigued by their pipeline and also ROR2 stuff.
Oh right.
There's
Okay.
someone doing that. I'm not really sure who, but
Yeah. So for the audience, MBE has a ROR1. I'm just gonna go with ROR1 because it rolls off the tongue better. In phase one, and it's targeting TNBC and lung ovarian cancer. It's actually interesting that they're targeting ovarian and lung, not going for like the MCL, CLL types. So like maybe yeah, like we've got maybe even more of a breadth of potential targets, like you sort of say, because it's expressed pretty much kind of everywhere, right?
it really is. And there's a couple of cancers that I haven't really seen pop up in some of the further stages of clinical development, like endometrial cancer and a couple other places where we've seen significant like RR1 expression. But yeah, it's just very exciting. I think in response to your point about MCL and why they're going after some of these solid tumors instead.
Yeah.
I think it's a bit risky, but I think the potential, if it works as well, and it seems to be they have a lead that they really believe in, I think the potential upside is just very ridiculous. So.
What have we got as like the prognosis? I need to get an exact number. Peter, have you got any numbers off the top of your head? I've been following you for a long time, so I know you're a smart guy.
Unfortunately, I haven't really followed this up very much, so I was just intrigued by the target. So I don't have anything to contribute on the investment aspect here.
Right. So, all right. Okay. Looking at the costs, right. So we've got ibrutinib, which costs, off the top of my head, on a quick Google search, is I used to work at Informa, where we would cover like therapies, like particularly rare diseases and CNS So we'd come up with like a patient-based kind of forecast, and I'm just gonna see how much this costs per year, because I'm trying to think like how much would each patient be worth if we were looking at it. Actually, do you know what? We've got STL on here. I'm gonna make him a speaker because I'm pretty sure he might have an idea. I just pinged you an invite, dude. See if it works. In the meantime, let me get a number up. Okay. No, that's not a number we want. Okay.
It's exciting as well that the potential MAP kinase pathway inhibitors that are just now being revisited. I think those are going to synergize quite well with ROR1, so I think they're both coming up at
the same time. Have you got any idea on like where they're at in terms of like phases for MAP kinase? Yeah, okay. I'm just taking a look at this MAP kinase, and there's going to be a lot of editing because of this Twitter Space headache. Definitely. How expensive is a Britanib? Okay. Wholesale price per month is about 13k for a Britanib alone. So let's say like ballpark number, we take that and then we apply that for a 12-month kind of number, and we say, okay, right, what's that? 113, 130 by 226, 156 as a rough ballpark. That's 156 grand per patient per year if they're taking it for the entire year. But I'm guessing that wouldn't be the case because they'd be taking it on a cycle, right? So do we have like an idea on the cycle length? Does it say? Got 4 weeks at assigned doses, and then there was nothing for. Yeah, there we go. So up to 4 weeks. Okay, so yeah, if we say 4 weeks of therapy at 13k as like a base number, and then we've got how many patients in MCL, CLL. Let's bring that up.
Did you say for MCL?
Yeah. Trying to get a number up. They always give you the incidence, but they never give you just like a boom, here's a number. This is how many people suffer from it, and I always have to do like a calculation of, all right, okay, this many per 100,000 people, right? Like, what's my total then?
Yeah, that was the hardest thing to do as well, because when you're writing a paper, like you want to emphasize the problem as the first line.
Yeah.
It's just so hard to get like accurate, up to date, like statistics and incidents and things like that. It's just.
Yeah, it'd be nice if they just gave like a point spot number of, okay, this is how much it is now, you know. But I think I read like it was something along the lines of what, like 86 odd thousand in the US. So if we go for like a penetration of about, I don't know, like as a high end number of like 10%, you've got 8,600 odd people there. I'm gonna open up my Python notebook because that should just make all its calculation stuff a lot quicker.
How accurate do your calculations usually end up being?
Well, when I was working at Informa, like I did the forecast for Vertex, so I know it's not cancer because that wasn't my specialty. and I covered cystic fibrosis, and touch wood, like my numbers were pretty good. Like, and I left the company. I looked back at the results after, and my mate was like, because she stayed as an analyst there, and I moved on pretty much after like 10 months. And they were like, yeah, no, your numbers are still holding up, because we sort of review it each kind of year. So I was like, oh, cool, like I got some decent numbers here. Um, yeah, but like I think with cystic fibrosis, it was a lot easier, right? Because it's a rare disease. Like there's smaller numbers, and I think the market then was so easy to model, just because, I mean, it didn't really have a therapy. Like there were no drugs, um, really for cystic fibrosis, apart from like the nebulizers that sort of just help, but they weren't curative or, um, I guess, like helpful in terms of like reducing, um, forced expiratory volume. Like your FEV1 wouldn't really like improve, so hardly anything. Um, let's get a number, 8,600. All right, let's just go with 13,000, because they'll do it for a full week, and let's say that that that's going to be like a peakish kind of number. I have, and I always got to count the zeros. 11, or I've got 111,800,000 as a number. That's not too shabby. And if we kind of like discount that back at like a 2% rate, and what's the market cap of ONCT right now? Let's take a quick look. I should probably just have Weeble like up. Weeble, if you want to sponsor me.
443 million, I think.
Yeah, so it's probably already starting to model in that number, but they've already got, what's their cash and cash equivalents. I think they had like a fair amount already.
Yeah, I was
Balance
reading
sheet.
an article about their burn rate, and it's fairly sustainable, to be honest. They could keep it going for 6 more years.
Yeah, they've got a decent amount. Like they've got, like, total assets here. Just looking at Weeble's numbers, they've got 118 million. Liabilities only at 5.86. So we can just assume like 113 million is just cash and cash equivalents kind of number. It's a fair amount, and they only, like, their cash flow was what, minus 20, minus 23, minus 40 thereabouts in 2018, 60 2019. So what, we're looking at like an average of like 20 mil. Yeah, 6 years. That's bang on. Sounds.
Now, if Johnson & Johnson or AbbVie acquire ONCT soon,
That would.
that could
Yeah, that's the next. We just need some buyouts, man. Like XBI has just been getting hammered. Like it always seems to just be like the turd of the crop when it comes to any sort of sell-off. They just get hit harder, and it's sort of just like, oh no, my portfolio is being fucked right now. Cool. All right. Okay. I think that's a fairly decent kind of view on Octonal, but just like switching up. I'm just gonna go like rogue here and switch up to something else that I saw on Twitter today. So I don't know if you guys saw this, but have you heard of Fama?
No, what is it?
Um,
Ticker.
So it's not actually a ticker. This is a company that, and the reason I'm kind of mentioning this, because it seems pretty fucking scary. It's a social media screening and automation and AI company, um,
Oh my.
and it 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 looks like this company basically gives like background checks on individuals when they're joining a firm, right? Um, and this guy, he applied for a job, and he got a 300 plus pager on all of his tweets. Everything he liked, like even the stuff that had been deleted, like these guys have access to. So like nowhere is safe. that that seems to be like the the main gist of it. Like I'm
Yeah,
right.
I mean, that's some scary stuff.
That's like it really is some
Yeah.
scary shit. Like I'm gonna bring it up real quick. One sec. Let me get this up because I saw this and I was just like, what the fuck is going on? Um, so all right, I don't like you're not a techie, right? Twitter usually they've got like an API
Yeah.
that you can use, but generally it only really allows you to like scrape a small amount of tweets, um, nothing big. And if you do want to try and get a bit deeper, then that's where sort of fun stops, and you kind of need to go speak to them. Um, so yeah, this company seems to have got like a private API with Twitter, and it literally screams Cambridge Analytica. So I'm just going to read this guy's tweets out. I had to get a background check for my job, and it turns out the report is a 300 plus page PDF of every single tweet I've ever liked, with the word fuck in it. Enjoy your dystopian bullshit. And then he goes, update. I came home to a package containing a printout of all 351 pages of it. Obviously the dystopia cares about wasting paper. But then he goes, on the plus side, I can now save these for posterity. And he's got like one of the tweets that's on there is like sharing a lift with a cheerful stone bunch who hopped in the back seat like I'm about to go chaperone a third grade field trip to the Nutcracker. Would you care for
Oh
some
my
vodka?
god.
And like that's what the season of giving is all about. Like this is, and then it has like it has a flag type and it says bad, and then it goes
You
flag
know what?
reason alcohol, and then it goes post type liked, and I was just like, what the fuck?
Yeah, my tweet's not that bad.
So I then the next part is like hilarious. He got another tweet and it goes, to this day, this is still the most big dick energy I've ever seen in a video, and it was a screenshot of this guy who basically he was on Who Wants to Be a Millionaire,
Yeah.
and he's got all three lifelines available. He's on $500,000 dollars, and he's got one question left to get $1 million, right? But he doesn't take any of his service lines, so cool tweet, right? And then the flag reason for this is language, bigotry, and sexism. What? What?
Yeah, there's a lot to unpack there.
I,
Yeah.
mate,
That,
I don't know. I just don't like.
yeah,
I find myself now like as soon as I read this, and I literally read this about 5 minutes before we jumped on this call. Um, I just I was
you
just like
try
I
to
can't.
censor what you say.
Yeah, no, I was just like shit. I can't like anything anymore.
I
I'm
mean,
I'm gonna go back to being a Reddit lurker. Like I'm just gonna read shit, and I'm never gonna like anything.
yeah. I mean, it was crazy before because on Twitter you didn't really like have things that people liked on your timeline.
Mm.
But now I can't like things that I actually like because it's gonna pop up on someone's timeline, and I've got all kinds of people following me. You know, I've got friends from high school. I've got people that follow stuff. I've got my supervisors. I've got people in academia. You know, I can't be liking some really, really stupid me,
Yeah,
but I want to do that. You know?
yeah, no, exactly,
Crazy
right.
thing.
This is just
It's a
crazy,
new age.
mate. He's then gone on and he's just like the especially creepy part is this didn't turn up anything at all relevant or incriminating. Um, so like there's some dude like okay like this guy's just applied for a job, but imagine like you got like these stalker types that are sitting in the FAMA. Maybe I should be quiet because I reckon like maybe they're just like I'm gonna be hit with like a
Listen, they've got this too. Yeah, they've
yeah.
got spaces.
They like some dude wants to. I mean, have you watched You on Netflix?
I've watched you. Yeah.
Yeah, so like you can get a guy like that who's working at FAMA and he's just like, right, okay, I want to find out everything about this chick that I've been I've been looking at for God knows how long, and he's got everything he wants. Like,
It's crazy. You know the White House actually does that as well. Like they archive any tweet mentioning the White House or the president or whatever.
oh really?
Yeah, it's a whole thing. One of my friends went on a date with this girl, and she was just absolutely out there.
Right.
But she essentially tweeted at Trump something really nasty. I don't know. The Secret Service showed up to her door.
No way.
Yeah.
What the fuck?
It's absolutely mental.
That is, mate. That's crazy.
Yeah.
That is definitely crazy. Yeah, I just had to get that out there because I was just like I've just seen this and I need to talk to someone about this because this is,
Sure.
mate, this is just nuts. I can't believe this.
So what do you envision as a kind of like the format for the future episodes of this podcast?
Yeah, so I think I haven't got like a really good structure down with it
I like
yet,
it, man.
obviously.
Spontaneous.
Yeah, but like I kind of just wanted to, like, I didn't want to be, I kind of wanted to be like the happy medium between, you know, like it's informative and you get some pretty smart guys on here to like talk about things that other people just don't have the time to sit and learn everything because there's just too much information, right? Um, and then yeah, we move into like sort of like evaluation aspect, and then just sort of like bounce ideas, and then finally you get to like a point where you just talk shit. Like there's always something in the world that's going on. Maybe it's not related to investing, and maybe it isn't.
Especially in these times, yeah.
Yeah, exactly, right. And I think, like, at least for me, like I kind of just I've watched like a couple of guys on like their podcasts and whatnot, and it all just feels a bit too kind of serious for me. And it's like, okay, cool. Like maybe that's their purpose, that you want to get you just want to get the information out, right? But
I've enjoyed this.
yeah, no, mate, it's
good
been
just chat, yeah.
yeah, no, it's been really cool. Like, thanks for jumping on, and yeah, you definitely made it pretty easy to go through.
I didn't want to sound so down on RR1 earlier. I want to keep people excited, so yeah.
We
Just wanted to point that out.
yeah, I mean, like, to be fair, you're saving yourself, right? Because like there's a lot of SEC hammers handing down like on guys that are supposedly pumping stocks because they're putting emojis on and then they're following up with a stock, right?
That's outrageous, isn't it? Like, they're not going to go after the guys on Wall Street. They've been doing this for decades.
Yeah.
And they're just going to come after this little guys on Reddit and just put rocket emojis on.
Mate, like, yeah, that's such a valid point. Like, I I see. Well, I did a bit of like Reddit scraping on Wall Street Bets, right? And like I was doing it for a period of time, and all of a sudden, like out of nowhere, like my files just became like a dictionary of rocket emojis, and that was it. Like I had no stocks anymore. It just became a redundant exercise because I was like, you know, this scraper's not bringing out anything useful anymore. It's just like pump it up to the moon, rocket emojis, like diamond hands and stuff like that. Yeah, no, literally that was it. And I was just like, dude, this is not. It's not helping me. I'm not getting anything out of this. So I moved over to just investing and biotech plays because they've got a couple of decent.
Yeah, that's a good subreddit.
Yeah. Thanks. I'm glad you endorsed me there because now I don't feel like I'm just just picking out random shit Reddits. Yeah, so that's sort of like where it kind of went. But with the pumping stuff, right? Like you've got Musk, just like you've got Musk like as an example,
Outrageous, really.
but then you've got like the YouTube guys. Like I don't know if you know KSI
Yeah.
and like all of those. Like those guys are just like tweeting like a crypto coin, like Polkadot. Like, is that
They get paid for it.
yeah? But is that pumping? Like you just said a word.
It has to be, right? I mean,
It's a word. Like, can you be like I say hello? Like maybe there's a hello ticker in a future time. Like, is that a pump?
I think, yeah.
Yeah, I have like a couple of thousand followers, and I just tweet and just say Cardano, just because I feel like it. Is that pumping?
I mean, it's gotta be. It has to be like I don't know if you know the YouTuber Marcus Brownlee.
No,
He
no.
does like tech stuff, but he exposed Tron.
Oh yeah.
Yeah. They sent him an email where will essentially pay you this amount of money to just mention TRX in a tweet, you know. And it was like, and it kind of framed it as like this. Oh, it's an opportunity to engage with other influencers and celebrities. We already have these people tweeting it. And actually, if you go look at their timelines, they did. I can't remember his name. I think Lil Yachty was one.
Oh right, mate. It's so funny you mentioned this because I have literally been seeing that guy. Like I haven't chosen to follow crypto, but Twitter have put this up like on my own feed. So like you get those little list things pop up and it's like Bitcoin or whatever, and then you just see all these tweets all the time. And I see Little Yachty just pop up 24/7, and I was like, "Where's?"
None of it is okay, but I
So,
don't know.
so Tron were paying him to essentially pump the stock.
Yep.
Wow, yeah. I've heard some pretty nasty shit with Justin Sun. Like apparently he's not just a bully. Like he's like a full on dictator, um, with his staff apparently. I'm just gonna bring up this article, Crypto News. It was on the 27th of Jan, 2020. And apparently the BitTorrent staff, like the headline just reads, alleged violence at Tron from boss Justin Sun.
Wow.
So, like I go into the article and it says that two former BitTorrent employees are suing Tron for 15 million, alleging they witnessed workplace violence where were harassed into product launches and experienced wrongful dismissal. And one of the things that happened was one guy saw two sets of shoes under a conference room door and then heard a loud commotion coming from the room. And that was followed by the sound of what seemed like a punch, slap, or strike of the hand. Then the manager was left with a red face and glossy eyes after the alleged attack. Like, what are you doing when your manager or your boss just slaps you?
Yeah. This is crazy shit.
Like, he makes Goldman Sachs seem like it's a lovely place to work at.
Yeah.
Weird guy. Cool, man. I think we're probably just gonna leave it there unless you got anything ONCT or ROR1 to talk about. That took a big diverse tangent.
Got nothing more to add, but great stock. This is not financial advice.
Yeah, it's not financial advice, guys. No financial advice here.
Sure.
Cool, man. Well, yeah, it was nice speaking with you, and hopefully,
It's a pleasure, man.
yeah, we'll look at the first half of 2021 with some more ONCT catalysts that are positive for us.
Absolutely.
Thanks again, buddy.
All right. Bye-bye.
Cheers, bye.